Good morning, everyone. Thank you very much for being here this morning. Thank you to Bionano for inviting me to give this talk. My name is Isabelle Raymound-Bouchard, and I am a clinical specialist in biological sciences at Hôpital Maisonneuve-Rosemont in Montreal, Canada. And I'm very happy to be here this morning to talk to you about optical genome mapping with CD138 positive plasma cells. I should mention that Bionano asked me to give a talk that's a little bit more focused on the technical aspects of working with CD138 plasma cells. So it will be a talk that's a little bit more technique focused, and then at the end I will talk a little bit about analysis. So we started working with OGM and CD138 positive cells a few years ago now. It started initially as a research project to evaluate if we could use OGM to replace fish as the standard method for multiple myeloma testing in a cytogenetics lab. We started with trying to develop and optimize a good working technical protocol that we could use with CD138 cells, as there wasn't one when we started. And then we also wanted to get used to and look at how to analyze what can be very complex OGM results in the context of multiple myeloma. So, and we published this paper a few months ago now in 2023. So if you're interested in getting more information, you can check it out. We then went on after the research project to do an actual clinical validation of OGM for multiple myeloma. We have validated or run more than 50 samples in total, which we evaluated for clinically relevant structural rearrangements and copy number variants and compared them to standard of care results, which is for us our FISH panel. We've also evaluated other parameters such as repeatability, precision, accuracy, etc. We had overall very good results, and we're very excited to be able to move into implementing this in a clinical lab as in our routine analysis. So we're hoping to be able to implement it in the clinical lab in the next few months, and we will be using OGM to essentially replace our FISH panel in the lab, with the exception of TP53, FISH for TP53 and 1P, 1Q. In terms of the technical aspects of working with CD138 positive cells, when we started this project a few years ago, we initially worked with de novo multiple myeloma, relapse samples as well, and smoldering myeloma. Our cutoff in the lab was for a plasmacytosis of at least 10% in order to be included in our analysis. And originally, when we started this, we needed to have at least 2.5 million CD138 positive plasma cells recovered to be able to move forward with OGM analysis. This is essentially because we needed to make sure we had at least 1 million cells for FISH analysis and then 1.5 or more for OGM. We are looking at now at potentially using less than 1.5 million cells for OGM, and I will talk a little bit more about that later. In our lab, we do a semi-automated purification or isolation of CD138 positive plasma cells using the RoboSep from Stem Cell Technologies, which uses an immunomagnetic positive selection. This can caused some issues in our runs at the beginning, but the beads thankfully can be removed, and I will talk a little bit more about that. So once we've purified our sample, we go directly to freezing. So we add DNA stabilizer directly to our sample, and then we freeze our pellets at -80. As I mentioned, we usually add DNA stabilizer directly to the total volume of cells that we want to freeze. We do this on ice, and then we aliquot into individual tubes. When we have greater than 40 microliters of volume, we add a centrifugation step to try and create as much of a dry pellet as we can and remove as much of the supernatant as possible. When we have less than 40 microliters, we don't usually do this because when we remove the liquid, we also tend to remove a lot of cells, and so it doesn't work so well. And then we freeze our aliquots at -80 until we're ready for extraction. We do not do any washing steps here, and this is simply because in integrating this into routine in the lab, people are very busy. There's lots of samples, so it's actually easier for us to do a straight freezing, removing some of the supernatant, and then we actually do a wash before we extract when we're ready to extract, after we thaw, At this point, we do a wash where we add more stabilization buffer to the pellet, and then we can transfer this to the low bind tubes, followed by a short spin to create our pellet. This minimizes the loss of cells during transfer to the low bind tubes, and then creates a cleaner, more visible pellet, and then we can continue with the extraction as per the protocol. So in terms of getting good DNA from CD138 positive cells, with the generation 2 kits that we use now, the latest generation of kits, 1.5 million cells is plenty to get enough DNA for OGM. And as I mentioned earlier, we're actually now looking at whether or not we can use 1 million cells for OGM, which is very exciting. We always elute in 55 microliters rather than 65. This gives us a little bit more flexibility. We usually have slightly higher concentrations, and we can always dilute down if necessary. We also homogenize for 72 hours always. We did try shorter homogenization times of 24 to 48 hours, but the success is somewhat variable. And working with myeloma samples, since these are not urgent necessarily, we usually find that 72 hours is better. And this fits very well with starting the technique on a Friday, and then you can come in on Monday and continue with the technique. As I mentioned earlier, bead removal may be necessary post-selection of CD138 cells. So the beads that are used during purification can cause blocking. And they can get stuck in the nanochannels of the chip before they're run on the Sapphire, and this can cause significant run issues and affect data. So this is an example of what it looks like when we have eluted DNA that's contaminated with the beads that were used for purification, and these get stuck in these pillar regions here, and then that's bad all around. And so we actually do this, we added a step of removal of beads during the DNA extraction after the inactivation of proteinase K. So as per Bionano's protocol, after we inactivate proteinase K, we actually add in a step where we place the tubes on a magnetic rack for 10 minutes. And then you can see here, these are the beads that are getting stuck to the magnetic rack. And it works very well, and then we can just remove the liquid, move it to a new tube, and continue with the extraction. We have tried playing around with different times, less times, different repeating 2 times 10 minutes, etc. But we actually find that 1 times 10 minutes is really best. It works very well. It removes a lot of the beads, and even if there are some beads left in the sample, which happens sometimes, they don't seem to cause any issues with the runs. So integrating this step, we get very nice runs. So there are some other important considerations that we have to think about when implementing OGM for CD138 positive cells. It can be somewhat difficult to get enough CD138 cells to do all the analyses that we want to do. The amount of cells that we recover obviously is highly dependent on the initial white blood cell count, on the volume, on the plasma cytosis in the sample, and it can be tricky to get enough cells. Also, if you intend to keep some cells for fish as a contingency or for a backup, it can be tricky to get the amount of cells that you need. So we have been looking and doing some comparisons to see if we can actually lower the minimum concentration of cells needed for extraction. And I will show you that those results in just a second. I will say that we're also looking at how we can find a balance between cell recovery and purification. So during the purification process with a RoboSep, there are a number of washes and steps that increase purification, but usually lead to loss of cells, and sometimes we get lower recovery as a result. So we're also looking at looking into how we can modify the RoboSep protocol to increase recuperation of CD138 positive cells, but still ensure an okay or adequate purification. So in terms of testing 1.5 million cells versus 1 million cells for extraction, we've actually tested 5 samples right now where we extracted both 1 million and 1.5 million cells and to see how they would compare. So you can see here our samples with 1.5 million cells extracted and 1 million cells, and you can see that our DNA concentrations across the board are actually really nice, with one exception here with one of the samples at 1 million it was just below the cutoff, but overall we actually got very nice results and it works quite well. When we run this on the machine, you can see what it looks like. So we have the same sample run 1.5 million cells extracted here in blue, and then this is the 1 million sample in orange. And you can see that overall the data looks quite similar. The runs are really nice. Both of them we get very nice map rate, very nice effective coverage, very nice N50 values. So overall, we're very happy with these results. It's very exciting, and it means that we may eventually be able to actually go to using only 1 million cells instead of 1.5 for our extraction in our OGM. And that means potentially running many more myeloma samples than we would have before. And I'm not going to go into the results in detail, but essentially for all the samples that I've looked at so far, for the 4 that we've looked at, for the RVA analysis, all the clinically relevant results are concordant between the 1.5 and the 1 million samples. So for the last couple of minutes, I'm just going to go over some of the results we had, or some of the interesting things we find from our RVA analysis. So overall, with myeloma samples, OGM performs very well with structural variants and CNVs in general. OGM helps to refine greatly and clarify the specifics of many of the abnormalities that we see, and provides much better context overall. This is just one short example. We had a case where routinely in the lab, our FISH panel does not include a probe for the T614 translocation because it's not usually associated with the worst prognosis. But we do T414, T1416, and 1420. And from the results we got for this example, we actually had normal results for IGH. So we had no idea that there was an IGH rearrangement. But when you look at the OGM results, you actually see a 614 translocation associated with a loss of chromosome 14. And in this case, the loss of chromosome 14 is actually masking the translocation by FISH probe. We don't see it, we just see 2 normal FISH signals as a result of the monosomy. So this was an interesting result, and kind of shows how OGM can really help better define some of the abnormalities that we see. And from our study, we did see overall a 30% increase in the prognostic yield that with OGM compared to just with FISH. OGM is also extremely interesting because it gives you a global visualization of myeloma genomes. And so you can see in the middle, really large scale chromosomal rearrangement events, which are very interesting. And it's going to be interesting in the future to see how we can characterize these, how we can better define them, and what actually this means for myeloma patients. Lastly, I will say that for the detection of smaller clones, and for cases especially where non-diploidy is suspected, there should definitely be a visual inspection of regions of great interest, such as TP53 and chromosome 1. And for sure, in non-diploid cases, great care should be taken in interpretation of CNV results, because non-diploidy can skew OGM data. And in those cases, FISH for TP53 and 1P1Q may be necessary. So I think that's generally what I wanted to cover today. I'd like to just thank Amélie Giguère, who couldn't be here to present today, but she actually started this whole project and got the ball rolling on many fronts. And then there was a whole team at Maisonneuve-Rosemont who worked on this as well, both the lab team and clinicians. And then Genome Quebec and Bionano also helped greatly. So thank you very much.